but that's the nature of the course, right? Most of it should make a lot of sense. I actually think this is a kind of a fun lecture, right? So let's focus and let's stop chatting. That is very distractive, right? And it's Friday, my voice is kind of going down, and I have to save it for karaoke night. Okay. Before we continue here with the tryptophan operon, do you have any questions on the LAC operon? Is everybody clear with the LAC operon? Yeah? Like what bacteria like to eat? It likes to eat glucose. So if it has glucose around, it's not going to metabolize lactose, right? If it doesn't have glucose, but it doesn't have lactose, then bacteria is in trouble, right? It's going to find something else to eat, but no glucose, no lactose means LAC operon is not going to be turned on, right? So in order for you to turn on LAC operon, you're going to need no glucose and presence of lactose. Remember, you have the CAP, the stimulating protein, right, activating protein that has to be bound to cyclic AMP in order for you to get good interaction of your RNA polymerase to the promoter. All right, so that was like summary of LAC operon in 10 seconds. All right. So here we are talking about this other operon. So LAC operon is inducible. You're only going to turn it on when you need to digest lactose, right? This other operon that we started to talk about is the tryptophan operon. So this is the example of the repressible operon, because you have it on all the time because you need to make tryptophan. But in case you have plenty of tryptophan available, you don't want to keep on making more tryptophan because that's wasteful. So in that case, you turn off your tryptophan operon. So this is the example of repressible operon, right? Now, we talked about the repressor protein before, right? And now we have to finish up talking about attenuation. And this is the part that gets a bit confusing because we are not very used to thinking about transcription and translation happening in the same compartment. But remember, this is happening in bacteria. So you have coupled transcription and translation happening. It means that one is right after the other in the same compartment because you don't have division of nucleus and cytoplasm, right? So here you have DNA, circular DNA of the bacteria. You have expression, you're going to have expression, continuous expression of tryptophan operon. And you are going to have, of course, the transcription going on. At the same time, you're going to have translation going on. So RNA is being made, RNA is being translated. Same compartment. That's the only way that this type of system can work, okay? So what is happening here? In the beginning of your tryptophan operon, okay, so here is showing your promoter region is over here, okay? So your promoter region is going to be over here. So here is the part that is getting transcribed, right? Transcribed part. So before you get to your genes for your tryptophan operon, before you get there, you have this whole stretch that is going to be transcribed into messenger RNA, like the DNA is going to be transcribed to messenger RNA. So here is showing you the transcribed messenger RNA, right? And this whole region over here is a region that is called UTR. And it's a region that was transcribed in the beginning. So it's the 5' UTR. And UTR stands for untranslated region. It means it's a part of the RNA that is not going to become protein. It's not translated. Okay? So this whole stretch over here, look what is happening. You have one stretch that is going to help to bind to the ribosomes. Remember, ribosomes are going to do the translation. You're going to start making protein, right? Remember, at the 5' of your messenger RNA, you have the cap, right? So you have cap proteins right here, and the cap proteins helped the RNA to get out of the nucleus, and it helped the RNA to find the ribosome. So here is an area close to the 5' cap that ribosome is going to interact, okay? In the meantime, where is your RNA polymerase? It was over here somewhere doing some more transcription, okay? So here binds your ribosome. So ribosome is going to start to go down this area, and this area here, it is called untranslated region, but it's a bit of a misnomer, but let's not get into too much details, okay? So let's see what is happening here. You are looking at the conformation of this messenger RNA right there, okay? So this messenger RNA has some regions, they are labeled here 1, 2, 3, and 4, some regions that have complementary base pairing, meaning that they can form hairpin loops. So you can form hairpin between 1 and 2, or 2 and 3, or 3 and 4. Okay, you can form several hairpin loops out of this. So the formation of certain hairpin loops are going to allow translation to happen, and then formation of other types of hairpin loops are going to block translation. So this is what is called the attenuation for the translation of these messages. Okay, so look what happens here. So this is the typical diagram that you see in every book. Okay, so you see here something like this. There is a squiggly line that has something that says tryptophan codons, and here this hairpin loop that is a kind of a double triple type of hairpin loop. It says 1 and 2 bind, 3 and 4 bind, and this one says attenuation. And then the other one says 2 and 3 bind, and this says the thing is going to go on. And when you look at this, it doesn't make sense. Like, I look at this and it's just getting me confused. And what I think at this point, I think, well, let me just memorize this. And if something like this shows up in the test, I'll just put the numbers in there and hopefully I'll get the answer correct. Okay, that's not the way to understand this. Okay, so this is why I put these extra slides here, so you can actually understand it. Okay, so principal idea here is that this is a type of operon that you can stop its working. So it's always working. Call it repressible operon. Why is it? Well, because you have to make tryptophan all the time. When you have plenty, you have to shut down the operon. That's the first idea. Okay. Now, let's look here at this thing. This is what the part that I like. Okay, and I put the link here to the video that you can go and watch this whole video if you want further explanation. Okay, so this is what is happening. Here is your DNA. Okay, here is your DNA. Here is your RNA polymerase. It gets in there and is making the RNA. So here is the 5' of the RNA, and this is going that way, but is synthesizing 5' to 3', so your RNA is coming out of there. Now, your RNA polymerase is going to keep on going. Okay? Now, right after your RNA starts to be formed, now comes your ribosome, your different ribosomal subunits, your small subunit, your large subunit. They interact with this ribosome binding area in your messenger RNA, and the ribosome starts to make protein. So here is a protein coming out of that ribosome, big subunit right there. Okay, so this is a protein coming out, and ribosome is going along. And then the ribosome comes to this section here, and this is where you have 2 codons for tryptophan. Okay, so as we're going to see later in more details, when you do translation, your ribosome is reading the codons, is bringing in tRNAs that have the amino acids, right? So the amino acids get here to these areas in the big subunit, and then you form your peptide bond, because your tRNA brings in your amino acid, and tryptophan is an amino acid. And here is a codon for tryptophan. Everybody clear with that? Yeah? Okay. Now, let's say that you don't have enough tryptophan around. Okay? You don't have enough tryptophan around. So when ribosome gets to this area right here and reads codons for tryptophan, it's going to sit there and it's going to wait for that tRNA to bring in the amino acid. So it's going to sit and wait, but you don't have enough of it around. but it needs tryptophan. It's not going to move if it doesn't have the tryptophan. So it's sitting and waiting, you know, hey, you, tRNA, wait, tryptophan, are you around? And then tRNA is like floating somewhere else because there's not a lot of it, right? It's like, oh, it's my job, I'm coming, hold on, I'm getting there. So ribosome is waiting, come on, I'm getting there, I'm bringing tryptophan. But you gotta wait until I get there, I'm getting there. In the meantime, what is happening? You have your RNA polymerase that keeps on polymerizing, right? So at this point, all this big stretch of RNA was made. So now you have these areas that can form a loop. Okay, so you have segment 2 and segment 3 that they can form this hairpin loop and interact. Okay, so by the time RNA polymerase gets to the segment 4, this loop here is already made. Okay, and this loop here has a conformation that allows the ribosome to keep on going around it and keep on making the protein. So this is a signal that says keep this operon on, keep making more of these tryptophan enzymes because we need more tryptophan. We don't have plenty of it around. Okay, so when you see this hairpin, the interaction of segments 2 and 3, this means make more tryptophan. Keep the thing on, right? Now let's see this other situation in which you have plenty of tryptophan around, okay? So this is a situation when tryptophan is high. So here, same start, same story. RNA polymerase gets in there, it starts to make the RNA, ribosome gets in there, it starts to make the protein, and then polymerase is going here, and ribosome is going there, bless you. And now, when it comes across these codons for tryptophan, it doesn't have to sit and wait because you have plenty of it around. So it goes very quickly toward, towards like catching up with the RNA polymerase. Okay, so when it's like right on the back, right following your RNA polymerase, now this is going to block this area in segment 2 before segment 3 gets to be totally polymerized. So now if you block segment 2, it is not going to interact with segment 3. Now what is going to happen when RNA polymerase is done, you're going to have this segment 3 and 4 that are capable of interacting with each other. So this is a signal that when your polymerase gets there, this interaction is going to dissociate your ribosomes from the RNA. So this conformation here is a stop signal for making more or keep on expressing that operon. Okay? Does it make sense? Yes? Everybody awake? Okay. Do you understand what's going on? Yeah. Am I going to ask questions about this? Yeah. Okay, the numbering has to do with how soon those sequences are made. This one is made first, that one comes after, this one comes after, that one comes after. So it makes sense to go 1, 2, 3, 4. Basically, yes. So you should know, and you see, as you see with the last test, I don't put a lot of drawings, but you are free to do your own drawings. You can write as much as you want and draw as much as you want on your papers, okay? So it helps a lot if you have that visual when you are taking the test. So do the drawing, put the labeling, and you say, okay, how do I know the numbering? Well, it's just sequential numbering. It should be simple. How do I know what's happening? Well, if you have this guy stalling around over here and this whole stretch being made, if 3 and 4 are made, here in this case, if 3 and 4 are made, then this is going to be at the end, is going to mean stop. Okay, so you have to understand what is going on here in order to know what to answer. Okay, all right, do you have more questions? Everybody's cool? Yeah.
The first part where tryptophan is low,
Yeah, it will get there. It's low, but it's not zero. It's never really zero. Okay? It takes a while because you do have tRNAs around, but they are not charged with the amino acid. Okay? So you have to interact with the tRNA that is bringing the amino acid. And you don't have a lot of amino acid around, so there are very few of those tRNAs that are available. So you have to wait for this movement inside of the cytoplasm and for diffusion to take place for the tRNA to get there. Okay, so you have just less of it. But it's going to happen. You'll never have just zero available, or the cell will die. Once your tRNA gets to your large subunit of your ribosome, that is when you can actually make your peptide chain and the ribosome is going to move. Okay, when we get to translation, we're going to see details on how the movement happens, which is also depending on GTP energy, equivalent of ATP. So we get that when you get there, you see more details on how that is happening. Okay, but it has to wait until the proper amino acid gets there. Right, cool? All right. Oh, finally, question. Get your iClickers ready. Oh, I forgot to put here those on. Okay. What is the purpose of the tryptophan operon? Okay, notice there is no choice E. Okay, thank you. Some people are a bit confused, so talk to your neighbors. Make sure you have the right answer. Does anyone need more time? No? Everybody cast in your answers? Okay, I'm going to stop it. What is your answer? D is to make tryptophan. All right, so now we get to the fun part. Eukaryotic gene expression. There's a lot of details here, okay? Now, when we looked here at your prokaryotic expression, you're mainly talking about operons, right? So you're talking about sigma factors and when you need a gene on, when you need a gene off, and it's kind of simple because everything is the same in the same cytoplasmic soup. Now, when you get to eukaryotic expression, you have a lot of levels in which you can actually control the gene expression. So you have in the level of compaction of your DNA. So if you have DNA that is very compact, you cannot really access the genes. That's one level of gene expression. And you also have at the level of transcription, right? If you don't have transcription factors around, you cannot do gene expression. If you have transcription factors that are sequestered in the cytoplasm, you also don't have gene expression for those transcription factors. Remember the example I gave you for interferon, that you have transcription factors that are called STAT, like STAT1 sequestered in the cytoplasm, right? So if your transcription factor is in the cytoplasm, it's useless for gene expression because it's not in the right place, okay? So regulation of transcription can be at that level of sequestration. If you don't make transcription factors, you're not going to have expression, right? You can have expression on RNA processing. Remember that you can have some issues with RNA if it doesn't get processed properly, like splicing. RNA can get degraded. You can have also alternative splicing. That's when you can have isoforms, right? Over messenger RNA, you can get isoform proteins, right? So lots of expression control here. And you can have the stability of the RNA. that can be different. Remember, the bigger the poly-A tail, the more stable the RNA, the longer it's going to hang around in the cytoplasm, the more protein you're going to get out of it. So that's another level for control of gene expression. Okay? Then you can also have proteins that are held in inactive form, just sitting in there in case they are needed. Okay, although the protein was expressed, it's not quite functional, so another level of control, right? Then you can also have control at the level of modification of active proteins, like for example in phosphorylation. Sometimes phosphorylation can activate, sometimes it can deactivate. So you have levels of protein function that this also falls into control of gene expression. Okay, so many levels in which you can control how much of the gene is going to be expressed and how long is going to be expressed and the level of protein you're going to get. Okay, and we're only talking about proteins. We're not even starting to talk about RNA functions, other RNA functions. Okay? All right. So here are levels in which you can control gene expression, as I mentioned. Okay, I just mentioned that. RNA export is also another level of control. And I'm not going to get into details on this thing because in previous years I have people who started to complain about the research and how mRNA export is going, blah, 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 blah. Just give you a brief idea. Certain viruses block messenger RNA export of the cells that they infect. For example, influenza virus. Okay? Influenza virus. When you get a cold, when you get the flu, it is going to block mRNA export in the cells that the virus is infecting. But it's very tricky. It blocks host messenger RNA export. It doesn't block its own messenger RNA export. Virus is very fascinating. Okay, so this is one level of control as well. Okay, then of course you have translation and protein activity, protein degradation. Right, when you add tags to protein like ubiquitination, those tags are telling the protein that it's time for it to die, to be degraded. Okay, so that's another level. Okay, all right. And also you're going to have, as I mentioned, not only degradation of the RNA, but you can also have inhibition of the RNA. That inhibition can happen because you might have small RNAs or inhibitory little pieces of RNAs that can bind to your messenger RNA and stop its function. Okay? Sometimes you can have small inhibitory RNAs that are coming from other cells that want your cell to stop function. Sometimes you have these little RNAs that are coming from your gut bacteria. Yeah, there are regulations are coming from everywhere. Okay? All right, so here. Factors that are important in control of your gene expression. Okay, you have these factors that are called trans factors. What it means is that it is not in the same DNA molecule that you find your gene, or it could also come from outside of the cell. Okay, so you have a lot of these factors. And of course, this is only to do with eukaryotics, okay? Eukaryotic control, okay? So a lot of these trans factors are proteins that are made from a different chromosome, or it could also be RNAs that are also coming from a different chromosome, and then it gets to this chromosome for the gene expression and interacts with it. If it is able to stop the gene expression, so it is a negative control. Most of the time, trans-acting elements are activating gene expression. Okay, so this whole idea here is that this molecule is coming from somewhere else and is not in the same chromosome molecule in which you are having this gene expression. Okay. So examples of trans-acting elements, those are transcription factors, okay? They can either activate or inhibit the transcription, okay? You can have repressor proteins. You can have activator proteins. And here is an example of what it would look like in case you have this activator protein. So here you have your DNA molecule, okay? This activator protein got expressed somewhere. It got to this place, and now is going to interact with a part of the DNA that is going to bend, is far away from your gene, okay? Is going to bend and is going to allow interaction of this activator with your transcription factors. Remember these guys over here? TF2D? Remember TF2D? They has TATA binding protein, okay? So these guys over here, when this activating transcription factor interacts with transcription factors that are already there at the promoter, then you can enhance gene expression, okay? So let's see what is happening here. What type of proteins can do that? You can have this, ubiquitous protein, or you can have some protein that interacts with histone acetylases. Remember, if you put acetyl groups on histones, what happens? Yeah, so that information is not going to disappear. You're going to need it later, okay? So this protein over there is bringing acetylases to open up the DNA, okay? You could have some other protein that is bringing chromatin remodeling complexes that also help to open up the DNA. And these proteins here that were made somewhere else, those are considered transactivator factors. On the other hand, you can have repression factors that can get in there. Okay, so here is a protein that is able to interact with a piece of the promoter and block the interaction of this activator to the promoter. Okay, so this type of repression is called competitive blocking. Blocking of what? Of the DNA. So you have here your promoter. So this is the DNA. So this whole name of this is competitive DNA binding or competitive DNA inhibition. Okay. Now, besides just being competitive for the same site, you can have repressor proteins that bind to a site and then interact with an activating protein in order to stop that activating protein. Activating protein had its own little different site to bind, but if that guy binds over there, it blocks the activating protein, and then you don't have your gene expression. Okay, so this is no competitive, but is more specifically called masking the activation site. Okay. And then you have another form of repression in which your repressor factor binds to your initiation complex, to the TF2D, and blocks the RNA polymerase II from starting the transcription. Okay, so these are all forms here of blocking the transcription. Okay. Besides that, what you can have here? You can have repressors that interfere with chromatin remodeling, okay? Meaning that it can kick out the chromatin remodeling and cause compaction of the DNA. So that's another way to block transcription. Or you can have repressors that bring in deacetylases. So that is also going to block transcription because you're going to have compaction and formation of those nucleosomes, right? So here you have 5 different forms of repression of transcription based on those controlling proteins. Okay. So those are your transcription factors. In comparison, you can also have cis-acting factors, right? So cis-acting factors are going to be in the same DNA molecule as your gene that is getting expressed, okay? So most of the times, you're going to find that these factors are DNA stretches, just stretch of DNA, okay? But the difference between this and the trans is that this one is going to be in that same DNA molecule. Okay. So examples here of cis-acting factors. You can have promoters or enhancers. Okay. So a promoter, you know, is a stretch of DNA. Enhancer is also a stretch of DNA that can fold and interact with your transcription factors elements. Okay. So enhancers can bend over. That stretch of DNA is going to interact with the proteins and enhance transcription. On the other hand, you can have silencers that interact with the complex and block transcription, right? And here you have the examples in prokaryotes that are the operators, but basically for eukaryotes you are concerned with these three over here. Okay. All right, so regulatory protein complexes is basically with the same theme. Okay. So you have some proteins that can activate directly or some proteins that can repress. Okay. So you call those proteins either co-activators or co-repressors. And the name basically explains what the thing is doing. So here is a slide that is summarizing these points, okay, on regulatory transcription factors, and basically is just going over what I had already mentioned before. Okay, remember that you have certain genes that are at a basal level. Here you're really talking about the genes that can be regulated either up or down. Okay. And here you have a visual of what an enhancer is going to look like. So look, here says enhancer. Where is the enhancer? Enhancer is a stretch of DNA right there. Okay? So the enhancer is a consensus sequence. It is evolutionarily conserved consensus sequence. So here what happens is that when you have assembly of your transcription factors at the promoter of the gene, you can have this protein that is part of your complex of transcription. This protein is able to interact with a piece of the DNA that is far away from your promoter, and this sequence of DNA enhances the protein to push polymerization, to push RNA polymerase II. Okay, so enhancer is a piece of the DNA in the same DNA molecule. Okay. All right, so also for control of gene expression, you can also have these genetic switches, okay? When you have genetic switches, you can either have negative control, like turning a gene off, or you can have positive control. The gene is off, you turn it on. Usually for eukaryotes, you tend to turn genes on, not really turn genes off. Okay, so usually when you're talking about genetic switches is when you turn the thing on. Okay. So here you can read on your own. I want to get to the interesting part. Okay, this is very much intuitive. All right, so this part here, when you have positive or negative control. Okay, so keep in mind, usually with eukaryotes, you're going to have positive regulation. Okay, but here are examples that you can have either negative or positive. So let's see what happens here. When you look at negative regulation, what it's telling you is that the gene is generally off when you have this protein that is bound. Okay, so this protein here is doing the negative regulation. It's sitting there and keeping the gene off. Okay? In order for this gene to be turned on, you're going to have to come with another factor that is going to pluck this thing out of there. Okay, so the job of this protein over here is to keep the gene off. That's the negative regulation. Another example of negative regulation is when you have the protein with some kind of factor that is sitting in there. In that case, breaking down that factor dissociates the protein and then the gene turns off. but the presence of this thing over there keeps the gene off. So when you see the presence of something keeps the gene off, that is negative regulation. Okay? So in comparison here, when the presence of something turns the gene on, that is your positive regulation. This something protein can do it on its own. Sometimes that something protein needs a cofactor. Mainly when you see the thing goes to the promoter, turns the gene on, that is positive regulation. Okay? All right, so make sure you understand what this picture is telling you. Okay? All right. So here a bit on details on how is it that the regulation is done. And I had mentioned before that you can methylate the genes. We had looked at methylation of histones, right? Remember histone methylation? If you add a methyl group to your histone protein, you're going to stabilize the positive charge. If you add a lot of methyl groups, you really stabilize the positive charge, right? Now, this is the same type of comparison of adding methyl groups and inhibiting gene expression. So when you talk about methylation of DNA, you are usually talking about inhibition of gene expression. Okay? So there is one nucleotide that can be methylated, and that is your guy cytosine, which is right here. Is this a purine or a pyrimidine? Pyrimidine. Okay, that's a pyrimidine. It's a short guy, only one ring. Remember Napoleon complex? It's a little guy with a big name. Okay, this is a pyrimidine. And this guy is cytosine. So cytosine can be methylated. Okay. Now, in order to get a methyl group on your cytosine, you have specific enzymes that, thank goodness, they have a simple name. This thing is called methyltransferase. What is it going to do? It's going to transfer a methyl group to cytosine, right? Methyltransferase. Okay. And you're going to have here your CG interaction that is going to be recognized by that methyltransferase. All right. So here, how is it that your methyltransferase is going to work? This is the part that starts to get interesting, okay? So here you have your enzyme, DNA methyltransferase. A lot of times you see this abbreviated as DNMT, okay? It's just a DNA methyltransferase. What is it going to do? It is going to use a molecule that carries a methyl group, and is going to transfer this methyl group to your cytosine, okay? The molecule that carries your methyl group, that is your friend SAM, okay? When SAM is carrying here the methyl group, then it's charged. When it does not have the methyl group, so it is oxidized, okay? So then you have your SAH. All right. So a little bit more in this detail over here, okay? Who is SAM? Who is SAM? S-adenosyl methionine. What does that mean? S for sulfur right there. Adenosyl, because look, it has adenosyl right there. This thing is a nucleoside, because it has your nitrogenous base, which is adenine, right? It has a nitrogenous base. It has, what is this thing here? Ribose ring, but it doesn't have a phosphate, okay? So this thing is S-adenosyl methionine. It is charged with a methyl group right there. So that is SAM. It's very important for your metabolism throughout the body, okay? Is the most important methyl donor that you have, okay? So SAM gets in there and donates a methyl group, as we've seen before, to a cytosine, but that's not the only molecule that can get methylated. Yeah? Sorry? What is it on the left? On the top here? COO minus. This is your carboxyl group. So let's do this. 1 second. So if I color it over here, okay, hold on. Okay, it's not happening. Okay, so I'm just going to write on top of it, okay? COO minus. This is what it is. But phosphate is not there to begin with. So this is a different thing. It's not a nucleotide. Okay? Yeah, it has the shape that resembles a nucleotide, but it doesn't have a phosphate. Okay? So it has this sulfur that is charged, okay? And it has a carboxylic acid at the end over here. Okay? It kind of looks like a protein because it has amino group right there too. Okay? All right, so look what happens. Your SAM is an important methyl donor. It's not only important for DNA, right? But it's also important for control of other molecules in your body. Like, for example, when you have adrenaline that is working, right? You know what is adrenaline? Hopefully. Neurotransmitter, right? You have adrenaline working. You know, some people like to have adrenaline rushes, right? Same thing as epinephrine, right? So you don't want to keep adrenaline active for a very long period of time. So you can down-regulate the action of adrenaline by putting a methyl group in it. And SAM is the guy that is going to donate the methyl group. So you donate a methyl group to this molecule of adrenaline and you can make it inactive. Okay? So SAM is important for a lot of methyl group transfers throughout the metabolism. Okay? All right. So how do you get SAM? Okay? You have this thing that is called a methylation cycle. You always have to have a certain level of SAM that is charged. Okay? You have to have always a certain level of SAM for your metabolism to be working at its optimal. Okay? Now, maintenance of SAM levels is nutrient intensive, is very sensitive to nutrients, to certain vitamins. Okay, look at this. What do you need in order to make SAM? Well, you're going to need folate, vitamin B12, okay? Look at this, vitamin B7, choline, cysteine, magnesium, methionine, niacin. It's a lot of stuff over here. A lot of vitamins. So how is it you're going to make SAM? Remember this guy over here? What is this guy over here? That is an amino acid that is very important also for starting to make proteins, right? So you're going to use this methionine in a protein complex that uses cofactors. Remember enzymes and cofactors? So cofactors over here, your vitamins, if you have plenty of those cofactors around, your enzymes are going to work well for you to produce charged SAM. And that guy is your important methyl group donor. Okay? When SAM donates a methyl group, it becomes oxidized and it is transformed into homocysteine. Right? Homocysteine gets into the cycle, gets converted into methionine, that gets converted to SAM again. If you do not have proper levels of your vitamins, you get stuck with a lot of homocysteine in your system. And that is bad. Have you heard of what homocysteine does to your body? High levels of homocysteine, it's bad for you. It's actually a marker for cardiovascular disease. High levels of homocysteine are correlated more in a causation form to high levels of cardiovascular disease. Okay? So here, homocysteine is the bad guy, right? So homocysteine is a marker for inflammation directly linked to coronary artery disease and heart attacks, right? Now, it's normal as you get older, much older than you are, let's say 50s, 60s plus, it's normal to have a bit of increased level of homocysteine But if you have all of your vitamins in check, you should not have too much elevation at that level, okay? If you smoke, you're going to lower your levels of methionine and you're going to increase your levels of homocysteine. That's bad for you, okay? How is it that homocysteine does the bad stuff? One thing that it does, it binds to copper and interferes with actions of some very important enzymes, okay? And besides that, you can have increased formation of free radicals just because of the presence of homocysteine, okay? Those free radicals can attack low-density lipoprotein particles and cause a lot of problems. And remember, if you go to the doctor for regular checkup, the doctor is going to check your levels of cholesterol, your levels of LDL and HDL, right? High density is a good cholesterol, low density is a bad cholesterol. If you have high level of homocysteine and high level of low density particles, then you are at more risk for cardiovascular disease, okay? And the main reason is because you're not using your homocysteine to make sand, okay? So keep that in mind. All right. What is the use of methylation? Okay. Involved in a lot of cell processes. Important for cell division. You can read this list on your own. But look at this. Early nervous system development. Immune system, immune cell differentiation. Neurotransmitter, epinephrine, acetylcholine. Okay. Histamine clearance. If you don't have good methylation levels, you cannot get rid of histamine. So you're going to have problems with allergies. Okay? Detoxification, biotransformation in your liver. Okay? Myelination. Myelination, what is this? Important for nerve conduction in your peripheral nervous system. Right? Also in central nervous system. And epigenetic control. Okay, specifically, you are controlling gene silencing. You're getting genes quiet by adding methyl groups to your cytosines. Okay. All right. So here, fun part about methylation. Okay. Remember that in order to get DNA, get genes quiet, you add methyl groups, right? So you have your methyl CpG island binding proteins that go there, and then you methyltransferase. And what that thing does is that it blocks gene expression. Now, you know that methylation of genes is cell-specific, right? So here you're looking at the situation that when you start to undergo cell differentiation, certain cell types are going to have methylation patterns. In comparison to other cell types, they're going to have different methylation patterns, right? So here, if you look at genes that are important for brain function, you're going to have methylation in certain chromosomes. You compare that to genes that are important for liver function, you're going to have methylation in different chromosomes. Okay? Now, when you look at cells that are important for gonad formation, okay, cells are important for egg and sperm production. And now you start thinking, what happens when you're going to produce a cell that is for reproduction, right? What happens when you produce an egg? What happens when you produce a sperm? What happens is that the cells that give origin to those meiotic events that undergo meiosis, right? That cell has all methylation cleared out because you have to start with a clean set of chromosomes for your new creation, for your new human being, for example, okay? So let's see what happens here, or animal or any living creature, okay? So let's see here, for example, what happens when you decide to use those principles of clearing up methylation patterns, and you decide to do some cloning, for example, right? We're talking about animal cloning, right? Everybody heard of Dolly the sheep? Yes? You know how Dolly was created? Everybody knows how Dolly was created? No? Okay, so brief idea of how Dolly was created. So for making Dolly, the scientists took the egg of the female, right? The egg, the head, the nucleus here with half of the amount of the chromosomes for the sheep, right? So scientists went there, plucked out the nucleus. So here is nucleus getting deleted, took out the nucleus. So you're only left with the cytoplasm and whatever is in the cytoplasm of that egg. The scientists went there and took a somatic cell from anywhere of this sheep, let's say from the skin, okay? Took a somatic cell, took the nucleus of the somatic cell, treated the nucleus with demethylating chemicals, meaning that it cleared up all of the methylation of those chromosomes, and put the nucleus inside of this egg cytoplasm. So you look at this nucleus and it has 2n number of chromosomes, right? So it has a full set, 2n, okay? All these chromosomes are demethylated, and they are in the environment that allows for development of the cell division and development of the embryo. Okay, so the nucleus has all the chromosomes that it needs for development of the sheep. Okay, and oddly enough, the thing worked, and Dolly was born, and you can clone animals, right? It's like as simple as that. Just put the DNA into, put the nucleus from DNA from the nucleus of a somatic cell into this germ cell, and you get a clone. Okay, so this is called somatic cell nuclear transfer. Usually, cells from the skin are used because they are easily obtained. Okay, now let's look at more details. What happens here? In theory, you get demethylation of all of the DNA, and that sets all the DNA for this reset stage. It's going to start from scratch. Okay? When the embryo develops, you're going to have, little baby Dolly here that is a clone of the mother sheep. Okay? Theoretically, everything is cool, everything works as perfectly as the science would allow you to have. Is that what happens? No, if I'm asking the question, probably the answer is no. What happened to little Dolly here? Very short life. And a lot of problems, health problems. Okay? So what actually happened is that instead of you demethylating everything in the chromosomes, what actually happens is that some of the chromosomes actually don't get demethylated. You don't have 100% efficiency of your experiments, ever. Okay? So some methylation stayed there. If you have failure for demethylation, you have certain genes that are part of that chromosome that are never going to be turned on. Because remember, methylation is gene repression, is regulation for downregulation of gene expression. Okay? So the cells that get differentiated and need the gene expression, they're never going to have what they need because the gene is off. Okay? And besides that, when you get the chromosome from a fully grown animal, the telomeres are shorter than the telomeres from gonadal cells. Okay? So baby Dolly, okay, the little sheep kid, was born with shorter chromosomes, meaning that the cell age was already the age of the adult, was not the age of a newborn. So that by itself is going to shorten the life of the sheep. Lack of total demethylation is going to cause health problems. It's going to cause defects on metabolism and certain cell differentiation. So that little sheep was really doomed from the start. Okay? Shorter life and health problems. a lot of health problems. Okay, and we're going to see some of these things that can be translated to what happens in some human cases. Okay, I'm not saying that human cases were cloned. No, I'm not saying that, but we're going to see what happens here. All right, so here, just to reiterate that methylation is a very important aspect of epigenetics, right? Hypermethylation means you slow down gene expression. Hypomethylation means you enhance gene expression. So if you take out methyl groups, you're going to have a lot of gene expression. A lot of times, hypomethylation in normal cells will cause them to become cancer cells. Okay? So when you look at genomic profile of cancer cells, you look at the metallome of cancer cells, you're going to see they are usually hypomethylated. Okay? So let's see here on these problems of DNA methylation or overall methylation. Okay? So here, how is it that methylation interferes, or what is the importance of methylation for health, right? As we mentioned here, a lot of cancers can come about if you have hypomethylation. Okay? Embryonic development also depends on methylation. Now, when you look at methylation patterns in brain cells, in neurons, you can see that different methylation patterns are related to certain tendencies of addiction, either drug addiction or alcohol addiction or even sugar addiction. Is sugar addictive? Yeah, you know how addictive sugar is? Huh? I heard you saying, yeah, actually, I got chocolate. It is Friday, you got time. This one is the one with good aerodynamics. Nuts or no nuts? All right, nuts. What'd you say? Okay, good. No. What do you say about the drug addiction? Is sugar addictive? How addictive? 5 times more than crack cocaine. Ready? Really? Woo, sorry, that was fun. 5 times more addictive. 5 times. Have you heard about rehab places for cocaine addiction? Can you imagine rehab places for sugar addiction?
Is it true that people will actually get violent if you take their drugs away from them?
Yes.
Like what if you tie them to a chair and then you burn their drugs and how would they?
That is torture, isn't it?
Would they like?
Well, I don't know. It never happened to me. So, all right. So, you have withdrawal effects. Anybody try to quit sugar cold turkey? Torturous. Yeah. Anybody try to jump into a keto diet? Have you heard of the keto flu? Not fun. If you cut sugar out of your diet, you're going to cut the carbohydrates too. You don't need, biochemically speaking, you don't need carbohydrates. Yeah. Yeah. Some berries are okay up to 18 grams of sugar or something like that. All right, so a lot of stuff happens with, sugar. What we call it is the other white powder. Okay, I can give you some stuff to listen to about sugar. All right. Psychiatric disorders, they have to do with methylation. If you have a good, healthy methylation, you are going to age healthily. So you can slow down aging. A lot of times you can stop aging. And sometimes you can even reverse aging. Yeah, I said it. It is possible. Okay, it's not the case of that, I forgot the name of that movie now, but not going to happen like that, okay? You know what I'm talking about? Long Button something, whatever that is. Who? Benjamin Button. It's not going to happen like that. Okay. But yeah, it's not totally scientific fiction, right? So here is when we have 5 minutes of the fun part of the class, right? When you talk about imprinting. All right, so this is another problem that nobody thought about, the cloning experiments with the animals. This problem of imprinting. Remember, Dolly, the poor Dolly, the sheep, got all of its chromosomes from the mother. And we couldn't even test what was all the mess up on this poor sheep's head related to what it did not get from the father. And that has to do with imprinting. So when you have the egg, you need the sperm in order to get a healthy baby. That seems obvious, right? Pretty much. Okay, so if you don't have your chromosomes coming from the mother getting together with your chromosomes coming from the father, you're going to have problems. Okay? That goes contrary to the idea that you only needed a full set of chromosomes. Okay? You might be able to get a living creature out of this, but it's not going to be normal. Okay? You need your set from the father, your working set from the father. That is 1N. You need your 1N working set from the mother. Okay? So this is when you have the different types of yeah. Yeah, you can do that. You can do all sorts of experiments. You're going to get a living creature the same way you got Dolly the sheep with only the mother's chromosomes. You can have half of a female with other half, 1N from another female, fuse them, and you get a living creature, but it's not going to be normal. Okay? You can also have certain, you can have babies cloned using, egg and sperm, but the cytoplasm from a different mother in order to avoid certain mitochondrial diseases. So you can have a baby with 3 parents, and that is, the baby comes out totally fine. And this is actually done in England. It's done in case you have certain diseases that have to do with mitochondrial dysfunction. Okay? Okay, so this imprinting has to do with inheriting the chromosomes from the proper parent. So this is when you have the cobra, the male cobra going hiss, hiss, and the female cobra going hers, I guess, hers. Okay. So you do have, mom and dad that have to work together to get a properly functional creature. Okay. Imprinting has to do with the function of all of those chromosomes that are not your sex chromosomes. So all the other chromosomes, you call them autosomal, right? So you need to have autosomal contribution from your dad and the autosomal contribution from your mom, because the origin of the parents is important for gene expression. That means the chromosomes that are coming with the sperm, they are not totally unmethylated. They come with some epigenetic factors that are important for development. Same thing with the egg. Okay, they come with their own epigenetic factors. So here we're going to look at 2 cases when a piece of either the mom or the dad chromosome is missing. So in this case, you have chromosome 15. So let's draw here. This is chromosome 15 from the mother, and this is chromosome 15 from the father. They should look the same, but they're not the same, because a piece of chromosome 15 from the father is missing. It got cut off, it got deleted. So big chromosomal deletion type of mutation. Okay? So now what happens here? First of all, how often does this happen? Unfortunately, quite often. Look at this. 1 in 30,000 births, this happens. Okay? So when the baby is born, you notice certain features, some phenotypes, like small hands and feet. Okay? When the baby is born, you don't know how tall it is, but when the child grows, you're going to see that the child is going to be short. Okay? Is not going to develop properly sexually. is going to be mentally delayed, is going to have a certain level of mental impairment. Okay? And most of the times, the person is going to be severely obese or morbidly obese. Why? Let's see what happens here. When the baby is born, there are certain characteristics to this baby. Like the baby is very floppy, doesn't have muscle tone. The baby, pick up the baby, the newborn, it's like a rag doll. It's like it does totally no muscle tone. You try to lift the baby is not lifting the head. A reflex of the baby, of a newborn, Apgar, reflexes. The baby is supposed to lift the head, and this baby doesn't. You try to nurse the baby, and the baby is not interested in nursing. You give the bottle, you try to breastfeed, forget it. The baby just doesn't want to eat. So most of the time, all the time basically, the baby has to have a feeding tube through the nose, otherwise the baby will starve to death. Okay? So the baby and the toddler is totally not interested in food up to about 3 years old. Then the problems really start. Around 3 years old, this toddler starts to get really interested in eating. And the child starts to basically eat whatever comes across. He's never satisfied. It never gets the satiety from eating whatever portion you give this child. Okay? And on top of it, the metabolism of this child is very low. The amount of calories the child needs is about 1/3 of the amount of calories that a normal toddler the same age would need. So this child is hungry all the time, and he doesn't even need all that energy. Okay, so obviously the child is going to be obese. And how do you handle a child with Prader-Willi syndrome? Well, the whole family has to work together, because you cannot leave food around anywhere. If the child will see anything that is edible, the child will eat it. Okay? And if you do not hide and lock everything that stores food in the house, the child can eat themselves to death. Very sad. Okay? So the whole family has to have total control of the food in the house. And of course, school settings, everything. Okay? And also, as I mentioned, a lot of times you have developmental delays. So the child doesn't even understand that he cannot be eating all the time. Kind of very, very sad. Okay. And on top of everything, most of the time the child is oppositionally defiant. Not only doesn't understand, it will fight you. Okay? All right. So mechanism for Prader-Willis. As I mentioned here, you have deletion of the piece of the father's chromosome, okay, chromosome 15. Sometimes you look at a karyotype of the fetus, right, and you don't see a problem, you don't see deletion, and you end up with a baby with Prader-Willis. So other things can happen. Another mechanism that you can end up with Prader-Willis is if you have hypermethylation of that arm of the father's chromosome. Okay, so that piece is useless. It could be as well deleted because it's not working. Yet another problem that you can have, look at this, uniparental disomy. That means the child could have inherited both of chromosomes 15 from the mother instead of one from the mother and one from the father. That will also lead to Prader-Willis. Okay, so everybody cool with these mechanisms? Yeah, let me see. Time is time here. All right, so I'm going to stop here just before angiometry.